Okay, so welcome to Life Science Cafe. My name is Julia LaValle. I'm a PhD student in the Neuroscience and Behavior program. This cafe Life Science Cafe has been established in 2011. It's organized by graduate student researchers at the UMass Amherst. Each month, we invite experts to share and discuss their research. Typically, we host these events in Amherst community, but we are continuing our efforts virtually. We would like to thank our funders, the Organismic and Evolutionary Biology Program, the Graduate Student Senate, and the Graduate School at UMass, the National Science Foundation, and our generous UMass Gives donors. Feel free to subscribe to our mailing list if you aren't already to stay up to date on all of our events. So I'm going to stop sharing here, and we're going to say hello to our latest speaker today. So we're very excited to have Dr. Dina Navon here to talk to us about her work on developing our understanding of the genetic basis of complex traits. Dr. Navon is a UMass Organismic and Evolutionary Biology alumna and currently an Inspire Fellow at Rutgers and a Life Science Cafe alumna as well. While at UMass, Dr. Navon both worked to plan and host the cafes and also gave a cafe on her PhD work a couple of years ago. So she's graciously come back to give us a little update on what she's been working on since. Thank you so much for joining us. It's great to have you here.
Yeah, it's great to be here. Thanks so much. I am going to start sharing, I think, maybe.
I'm going to send out a poll really quick first before you share the screen, just so they don't have too much going on.
Yep,
so right off at the top, we are going to have a little audience participation. So there are 2 questions, and we're going to be addressing these throughout the cafe. So I'm going to launch that. There should be 2 questions, so feel free to just take a moment to fill that out really quick. And I'll give you guys like a minute to do so. And I am going to send the link to the virtual sign-in sheet in the chat at the same time, just for those of you that are rolling in a little bit after I sent it out last time. Oop. There we go. Okay, almost got everybody. A few more people. All right, I'm gonna give you guys 15 more seconds to get in your answers. All right, I'm going to end poll for right now. Okay, so starting us off, we're going to come back to one of these questions a little bit later, but Dr. Navon is going to get us started with the cafe. Okay. So first question for you, Dr. Navon, that has to do a little bit with this, and might end up answering this question. Why do some organisms look different, even though they are the same species?
Yeah, that's a great question. So organisms might look different, even though there's the same species for lots and lots of reasons. But the one that we're mostly going to be focusing on today is plasticity, which is how the environment can actually change your appearance and behavior directly. Before I get too far into it, though, I just want to make sure that I'm screen sharing. Can you all see my
yes.
screen? Okay,
We can see your screen.
The title slide.
Thank you. Yes.
Yeah. Okay. Great. Yeah. So lots of things can sort of feed into what your appearance is at sort of an adult stage.
Okay. So some of those things, I know we're going to talk about a couple different definitions. So right off the bat, one of the things that we mentioned in the poll was a phenotype. So what can you define maybe phenotype and genotype for us, and maybe what the difference between the 2 is?
Yeah, absolutely. So your phenotype is the sort of set of traits that you and every other living organism has, right? So these are things that you can observe directly, things like eye color, height, the shape of your face, how well you tolerate changes to temperature and salinity. Those things are all going to constitute your phenotype. Your genotype, on the other hand, is your genetic sequence, right? So the sequence of G's, T's, C's, and A's and the order that they occur in. Um, so your genotype, I would say, helps create your phenotype, but it isn't the only input into your phenotype. It's one of the main ones for sure,
Okay.
but it isn't the only thing that affects your phenotype.
So in the first one, I'm going to share the results temporarily. In the first question in the poll, they did say the question was, can the environment around you change your phenotype? If it's not just the genotype, does that mean the environment is changing?
Yes, yeah, absolutely.
Okay. I think our audience did good on that one. I'm very proud of them as well.
They did great.
Good one. So what exactly is phenotypic plasticity then? I think we touched on that a little bit of what that means, but can you give us a little bit more of a definition of that?
Yeah, so for me, I would say that phenotypic plasticity is the ability of a single individual that has just one genotype to produce more than one phenotype in response to different environments. So it's a potential to produce more than one phenotype in response to these different environments. So I would call this kind of a broad category that encompasses many of those other inputs that I was talking about besides the genotype that feed into generating your phenotype.
I see. And does this occur at a certain stage in life? Are we just talking about certain edges of development, or can this happen throughout our life?
Yeah, it certainly happens a lot more when you're young, right? So your environment has a much more sort of extreme impact on your phenotype when you're developing. And so, but your phenotype can change at any stage. And with adults, sometimes a lot of what the phenotype is changing is actually the behavioral phenotypes. So for adult humans, right, when we get cold, that's an experience where we're having a change in our environment.
Okay.
When we get cold, we change our behavior, right? We go and grab a coat or a blanket or something like that. So I wouldn't say that phenotypic plasticity is limited to just one stage of your life, but it certainly, there's a lot more options for it when you're younger. But there's definitely phenotypic plasticity that exists in adults. And a lot of the research that I do actually looks at young adults, young adult fish, but young adults.
Indeed. But when we're talking about this, just quickly, are we talking about one individual responding and changing, or are we more talking about kind of on the side of everybody changing? And is this slowly everybody's getting used to it, or is it just a one individual changing throughout its lifetime?
Yeah, that's a great question, and it cuts to the heart of some of my issues when it comes to actually publishing my data. So in an ideal world, right, I would measure plasticity for every individual. I would clone each individual, I would raise them in two different environments, and I'd see how that changes their phenotype. But ethics committees don't really like it when I try to do that. Even with fish, and they don't care as much about fish, cloning fish, as they would care about cloning a human. I would not suggest, proposing to clone humans. But so because I can't clone fish easily or morally, I have to study plasticity in groups of closely related individuals. So because they're closely related, they do share a lot of their genotype, but they don't share all of their genotype. And so that definitely adds some squishiness to my data, right? There's some noise that gets added at that level, but no experiment is perfect. We have to do the best that we can to answer the questions that we're interested in using the technology and the moral code that we have.
Sounds good. Yeah. So touching upon our second poll result, I'm going to share it one more time. I think we stumped people a little bit more with this one. So a little bit of genotype versus phenotype here. So I asked them, what is an example of a genotype, and gave them different options such as hair color, freckles, eye color, skin color, all of the above, none of the above. So are any of these an example or of a genotype rather than a phenotype?
Nope, none of those are genotypes. Those are all phenotypes, yeah.
Yep.
So 54% of you all got that right though, which is pretty good, because I think it was kind of a trick question.
It was. We're mean sometimes, I'm sorry. So then I do have a little bit more of a definition question one more time, because it seems like we're touching a little bit about on what exactly evolution is. So what is evolution in your definition? I know that's a very broad question.
Yeah, so I define evolution as the change in a population's phenotype over time. So an individual can't really evolve because that time component isn't quite there. When we're talking about evolutions, we're talking about changes over generations, and an individual doesn't encompass more than one generation.
Okay.
So that's different from plasticity because plasticity is that change in an individual.
Okay.
Yeah.
All right. Thank you. That just makes it a little bit more distinct. I can see where that difference is. So one of the things that we talked about is how environment can change phenotypes, but what are some of those ways that we can see that change? What are some parts of the environment that really have a strong effect on our phenotypes?
Yeah, well, plasticity that effect of the environment can be really, really drastic, or it can be more subtle. And so I do have some examples that are more specific, trying to give you sort of a concrete example from the title slide. So here we have Daphnia. So these are planktonic crustaceans. And these two individuals that you're looking at here are phenotypically very different, right? We have our animal on the left, which has these long spines on its head and its tail, and we have the organism on the right, which doesn't have a spine on its head at all and has a much smaller spine on its tail. And what's really cool about these organisms in particular, they're great for plasticity research because they actually clone themselves.
Okay.
And yeah, and so these two individuals are actually clones of one another. And so the only difference between them is that they were raised in different environments.
Okay.
And so the environment that the one on the left was raised in was an environment with a lot of predators. And so they grew up sort of sensing those predators in their environment. And so they grew these long spines, which helped them kind of ward off their predators. And the one on the right was sort of raised in an environment that didn't have any predators in it. So that's a pretty drastic example, right? Where predation, whether you have it or not, can really impact your phenotype.
Yeah.
Another pretty dramatic example of plasticity here is in spadefoot toad tadpoles. So these are two, they are not clones of one another because tadpoles don't really do that, but they are closely related. So they are probably siblings, I think, of each other. And so the one on the left, again, is much smaller compared to the one on the right. They were raised at the same time. So this is not just like an age difference or a developmental stage difference. These were raised pretty much identically, but the one on the left grew up in an environment that didn't have a lot of other tadpoles in the environment. So it wasn't really feeling a lot of competition from its siblings. There was plenty of resources to go around.
Okay.
The one on the right here was raised in an environment with lots of other tadpoles. And so it was feeling that competition pretty heavily. And because it was already on sort of the larger end of the spectrum when it came to its size, it underwent this really rapid increase in size, even beyond where it was already at. And it changed its gut composition a little bit, and it actually started to prey on the other tadpoles.
Okay.
So it switched to carnivory to kind of try and escape that competition from its siblings.
Okay.
And so again, this is the environment having a really pretty major impact on what you wind up looking like at a later stage.
Okay. So can I
Yeah.
ask a follow-up question? So there's clearly there's interactions with other organisms that influence this. Is there anything for like temperature, anything along those lines? Is there something in the actual that's not related to other species that can, or other individuals that can also affect this?
Yeah, absolutely. The temperature one's a great one to bring up. I know that for some reptiles, if their eggs are raised at a certain temperature, they become male, and if they're raised at a different temperature, they become female. So that's a pretty dramatic one.
Yeah.
Yeah, all sorts of things like that can absolutely impact all sorts of aspects of your phenotype.
Awesome. Okay. So how does studying this phenotypic plasticity help us understand how a species will respond to like environmental and climate change as this, like, I know we're in a bit of a crisis right now. So can you get an idea of how things might shift because of how you're looking at this current shifts?
Yeah, absolutely. So one of the main ways that plasticity can help us sort of understand and predict how species might respond to climate change is that um, plasticity we know can permit populations to persist in the face of sort of a new or rapidly changing environment. Because organisms that are lacking in plasticity might not actually even be able to survive that abrupt change, but with plasticity, they might have the flexibility to survive long enough to evolve to coexist with that change.
Okay.
So that's definitely sort of one of the main ways.
Awesome.
Yeah.
Okay, thank you. I am going to now open it up to our audience. If anybody has any questions, feel free again, just a reminder, you can either type it in the question and answer down at the bottom, or you can raise your hand and we will unmute you so that you can also speak verbally and we can just interact directly. Either one is perfectly fine. Just get my bubble open too so I have it ready. Multiple windows difficulty, you know.
It's a really different format from my last cafe.
Yes, it is a slightly different world we're living in right now.
Just a bit.
I do have one question that has come up really. So a little bit of a question, I think, harkening back to our discussion on evolution. So how are these organisms responding to these environmental triggers? Is it next generation change? Are they dying off? Is the individual changing as the environment change? What are we seeing?
Yeah, I mean, within sort of the broader context of reality and biology, all of those things are absolutely happening. For me, plasticity is mostly that individual change. They're gonna very rapidly respond to whatever environmental change they're experiencing.
Okay, okay, thank you.
Yeah.
Another one, hold on 1 second, let's see. Got one that's going to be, we're going to address one in the next column, so I'm going to wait off on that one. I think we're okay for right now. So we're going to move on to the second cafe. And again, if you want to ask some questions, feel free to type it into the question and answer or raise your hand. We're going to have another panel, but I'm going to keep this one short because we have one that's addressed in the next one. And we'll let you guys ask again next time. Sound good? Okay. So my next question to you is going to get a little bit more focused in on your work. So you study fish specifically. You mentioned that up at the top. Why fish? I kind of is the question. Why are they a good model to study phenotypic plasticity? What about them makes them special in this regard?
Yeah, fish are really just a great model system for lots of different kinds of research. They're vertebrates, so they share a lot of their DNA with humans. They have a really short generation time, and they're going to produce a lot of babies, which is really important if you're doing, sort of genetic manipulations and those kinds of things. You want to have a lot of embryonic material, for lack of a better term, a lot of babies that you can split up into different treatments and all that kind of stuff. So they're really great because they do produce lots of offspring very fast. And compared to other model systems like mice, they don't take up as much space. They're relatively cheap to maintain, like their food's a little bit cheaper. So those are some like logistical reasons why fish are really great model systems. More specifically for plasticity research, there are a lot of species that are very plastic. There are some species that aren't very plastic. And so we have that sort of variation that we need to really dig into what actually makes an animal plastic or not. And so for me, that's really a driving question behind a lot of my research.
Awesome. Okay. So what sort of fish specifically do you work with? I think, and what did the last time we checked in? So you've worked on 2 different species of fish so far or more at this point?
I've worked on I would say three different groups, and I do have a slide for that. So let me skip through a couple of these because we're just gonna ignore all of that. Yeah, so my undergrad project actually started out looking at 3-spine sticklebacks. So these are a species of fish found mostly in northern temperate climates. You can find them in Massachusetts. You find them a lot more in like Alaska and British Columbia. And so the research that I was doing on those guys was actually looking at reproductive decisions. So for a female, how big her eggs were, how many eggs she was going to produce. For a male, how big his testes were, and that kind of thing. And so I use stickleback to answer some of those questions, and how different populations of stickleback in different lakes with different environments were changing those reproductive decisions. So still sort of in line with plasticity, but not very specifically about plasticity. In my PhD, though, I switched over to a combination of African cichlids, so like this buddy over here, and zebrafish. And so African cichlids are a really cool group of species. So they're pretty stunningly diverse. There's lots of changes in their color patterns. Different species have different colors. They also have different other kinds of traits, like they change their face shape based on what they eat, all that kind of fun stuff. But there's a lot of species of cichlids. So there's like 3,000 species.
Wow.
And a lot of them live in Africa. And so one of the main rift lakes in Africa, Lake Malawi, is where we get a lot of our cichlids from for the research that I was doing.
Okay.
Zebrafish are a little more uniform, so they don't have as much variation in their phenotype, but they are super well established models for development and genetics. So a lot of the embryology for vertebrates was worked out in zebrafish first before they moved on to like chicken and mice and stuff like that. Same with a lot of the genetics tools. So we have a lot of those molecular tools like genetic manipulation, using CRISPR-Cas9, for example, and that kind of thing, very well worked out in zebrafish. Whereas in cichlids, that sort of thing is only just sort of starting to gain a foothold.
And just to interrupt you really quick, CRISPR-Cas9, can you just tell us really quick what that is for people who don't know?
Yeah, it's a way of engineering your genome. So it's a way of actually manipulating the genetics of an organism. You can inject them with these molecules at a very young age, at a single cell of development, and sort of change how their genes are being expressed
cool.
as they develop. Yeah.
Thank you.
Really cool. But so they have very different strengths, right? You have this natural variation going on in cichlids, and the really amazing molecular slick tools in zebrafish. And so my PhD advisor wanted to build his lab by leveraging that complementary strength, to do studies using both cichlids and zebrafish to explore how the genes and the environments are working together to shape your adult phenotype.
Yeah.
And so that was my PhD, which I defended in 2019. Then I started a postdoctoral project at University of California, Riverside, working in collaboration with someone up at the University of Calgary. And I went back to stickleback, actually. And I was looking at how their feeding behavior changes with their different diets, and how that differs between populations. So I was doing a lot of high-speed video capture. Yeah. And so now, since the funding on that project ran out, I am back to working with cichlids and zebrafish to understand plasticity, trying to fill in some of the things that I didn't get time to do in my PhD. I'm now trying to do as a postdoc.
Awesome. That sounds great. So that's a lot of different things that you had to do throughout your career, and a lot of different projects, it sounds like, that you've been able to take on. So what does a day of research look like to you? Are you going into the fields, getting fish? Are you mostly in the lab? What does it look like to be Dr. Dina Navon for a day?
Yeah, it's definitely been different at different times in my career. So for right now, like, I'll answer that based on what my actual day looks like right now. A day in the life involves a lot of working down in the fish room at Rutgers that I've basically built from scratch. So that was a lot of my first year, was figuring out how we could actually house all of my animals here. And so a lot of what I do is actually just the nitty gritty of taking care of my animals, making sure that they're healthy and happy and getting ready to breed. And then the rest of my time is kind of divided between actually collecting and analyzing new data, writing grants, and teaching.
Awesome.
Yeah. But all of my research right now, and this I plan on changing in the future, but all of my research right now is lab-based. So I'm going to manipulate something about these animals' environment, and then I'm going to look at how the phenotype is changing based on those different treatments. And so I'm also going to be linking how that phenotypic change in response to those different environments is linked to gene expression changes, and trying to get a better understanding of what genes let an animal be plastic or not.
Awesome. And so what are the environmental factors that you study specifically, and what are the responses that you're kind of measuring in your fish population?
Yeah, we've kind of touched on this a little bit. So a lot of my research focuses on how the diet actually impacts the phenotype. So if you're a fish, there are kind of two main ways of making a living and actually getting your nutrients from the environment. If you're living in an aquatic environment, you can either kind of feed benthically, so that involves being down on the bottom of the lake or the ocean or stream or wherever you happen to be living in water. You're down at the bottom interacting with the substrate. So you're scraping food off of a rock, you're sifting through the mud to find little critters that are living in the mud. You are getting up close and personal with whatever that sort of bottom of the lake looks like. On the other hand, you could also be up in the water column feeding pelagically. So that's just these open water column feeders. And so if you're doing that, you're going to be doing things like suctioning your food out of the water column. And so these are two great pictures of two different cichlids feeding in these different ways. So this is our benthic fish over here. And you can see that it's getting right up close and personal with that rock. And what it's doing is actually just scraping algae off of that rock. Okay. This pelagic fish, on the other hand, is kind of swimming around in the water column and suctioning food, presumably kind of microscopic food around here, into its mouth with suction feeding.
Awesome.
And so these different diets correspond to really different shapes. And so that's what I'm going to be showing you here. And it's true in lots of different species. So here we have two examples of Arctic char. We can also look at this in stickleback or in cichlids. And so what I want to point out here is that our benthic feeders are sharing phenotypes with other benthic feeders, even though they're actually more closely related to their pelagic counterparts. So those kinds of features that they're sharing, so if you're benthic, you tend to have a deeper body. You can see this really, really well on the cichlids, right? This is a much deeper body compared to this much narrower body here. I like to think about it as, do you look like a torpedo or do you look like a tank? Not to be overly militaristic about it, but this animal looks like a tank, right? You can't really move it easily through the water. If you tried to push it through the water laterally, it wasn't going to move very much. This one, on the other hand, looks more like a torpedo. It's a lot more slender and narrow-bodied. Other things that are changing, things like their jaws. Again, you can see this pretty easily on the cichlids here. So this benthic cichlid has a really short jaw. This pelagic cichlid has a really long jaw. And other things are also changing, like their fins and their body. I talked about their body shape, but their fins are also changing as well.
Okay.
And so the major driving question behind my research, like I kind of alluded to, is whether or not plasticity has something to do with these correlated changes to the phenotype. What kinds of genes are maintaining these responses as well? And so, yeah, that's a big part of what I do, is just trying to understand that. And so my PhD focused on the faces a lot because that's what my advisor did. Now we're trying to repeat some of those experiments, but actually look at everything behind the head. So your fins and your body shape. Yeah.
Cool. So were there any big challenges you encountered in your research? You can get your pick, maybe just working with fish, any behind the scenes challenges or steps that really you didn't expect?
Yeah, so I talked a little bit about how I started a project right after my PhD. So that was my first postdoc. And that was approximately 2 years of funding where I was going up to Canada and actually doing field research. And that project ran out of funding. And I found this out that I was running out of funding in a few months in May of 2020. So like many people, I was very much impacted by COVID and trying to figure out how I was going to find a job, secure my next position during COVID. and also sort of all of the logistics involved with that, because when I did finally get a job, obviously it wasn't going to be in Canada, it wasn't going to be really on the West Coast necessarily. So I actually did move across the country during the pandemic, which was definitely sort of a major challenge.
That's definitely a challenge, right?
Yeah, and so after all of that went down, right, and I was finally sort of settled in New Jersey with my fiance, who is now my husband, in October of 2020 was when we actually got here. The advisor that I started working with here didn't actually have space in his zebrafish facility for cichlids. So cichlids are significantly bigger than zebrafish. And because they can be pretty aggressive with each other, they actually need a lot more space, even than their size would necessarily suggest. And they also need a more complex habitat. So we actually use like broken flower pots and you know, smoothed off PVC pipe segments to create little territories for them to defend, that obviously takes up quite a bit of space. So for comparison, our absolute biggest zebrafish tanks here have about 1.5 gallons or so of water in them, whereas our absolute smallest cichlid tanks that are really only suitable for young animals have about 10 gallons of water in them.
Wow.
So just finding the space to get a room suitable for hosting that many fish was going to be another sort of big challenge to getting my research here going. And also doing all of this on a budget where, you know, unlike my advisor who had, you know, startup funds to actually build his fish room for him, I was doing it all on whatever scraps he can afford to send me. So that was the next kind of big challenge. And then another thing that I wanted to touch on here is that finding the right cichlids suitable for my research has been a pretty consistent challenge since I was really wrapping up grad school. So cichlid researchers in the US, myself, my advisor, and all of the other people like us, we buy a lot of our animals from the aquarium trade. So cichlids are a really common aquarium fish. They're sort of one of the first, you know, more challenging species that a lot of people would want to try. So, you know, you've got your betta fish and your goldfish, which are pretty easy to maintain. Cichlids are sort of a step up from that, but they're not so finicky as like a complex reef animal would be to try and keep in your house. And so we get a lot of our animals from the aquarium trade, animals that they don't want or that they're willing to sell to us for whatever price. And from where we're sitting, it seems like a lot of those aquarium fish are getting pretty inbred. So it's getting more challenging to import animals straight from Malawi. That's also true, sort of COVID brought another layer to that as well. And if you're looking to sell fry and your juveniles to hobbyists who are just going to keep them until they're adults and then they're going to die and it's going to be no big deal, they'll just go to the pet store and buy another one. That's not really a problem if they're super inbred, but it becomes a big problem if you're looking at these animals as sort of representative of the populations that you might find in the wild. And so we're trying to source.
Trying to look at phenotypic differences.
Yeah, and you're trying to get babies, all that kind of stuff becomes a lot more challenging when you're dealing with inbred animals. And so we've been trying to find, you know, ideally amazing would be if we could get some wild caught animals. But we haven't been able to find them. We have been able to find some, it's rare, of the first generation away from wild caught. So the babies of wild caught parents are being sold. And so I have some of those in the lab now, and they're actually just starting to more consistently breed. They're a bit small, but they're getting to the point where they're doing that. So that was another kind of challenge, was getting the animals, waiting for them to grow up, all that kind of stuff. When you're talking about a postdoc, which is a much shorter time period, like your PhD is going to take you 5 or 6 years, and you know that you're going to have that much time. Most postdoc fundings are closer to like 2 or 3 years. So anything that has to, you know, you have to wait for it becomes really nerve-wracking.
That's very fair. So one kind of follow-up from that is there's a lot of challenges that are involved in this. So what are some takeaways about your work that you want the greater community work to be excited about? I know that one thing we touched upon is how this could be important in the context of climate change, whether or not this could be something that you want people to understand a little bit more. What are your thoughts on what you think people should really think about this?
Cue in on. Yeah, absolutely. So my own sort of big takeaway from my own research is that, you know, when it comes down to that big quote unquote debate between nature and nurture, it's not usually one or the other. It's most often both of them. And it can be pretty difficult to actually tease apart how much of your phenotype is because of your environment and how much is because of your genes. And it can be very complicated. And so for me, that means we can't just fund study after study after study focusing just on genetics and expect to have a good handle on things like human health or human development. Mm-hmm. We need people like me and my advisors who focus and look outside the genotype and look at the environment as well, as much as we need people who are focusing entirely on the genes. But unfortunately, because our funding landscape right now is extremely competitive, it's much easier to just study the genes. You're much more likely, if you manipulate your genes, you're going to get a huge impact on your phenotype. And so those are a safer bet in terms of funding, because you're not going to get published if you don't have a big flashy result, right? And so that means that my grant proposals and the grant proposals of people who think like me have a much harder time getting funded.
Okay. That's a good takeaway. I think it really makes us think about what we prioritize in science and what maybe we should be encouraging and backing in science when we're looking at this sort of at a larger level.
Yeah, I think funding for basic research in general needs to go up. But even within what funds we currently have available to us, I think that the focus right now is mostly on genotypes, and that I think is to our detriment overall.
Okay. All right. Well, on that note, I think it's a good time to open it back up for another question and answer. So again, feel free to either type in the question and answer bubble down at the bottom, or you can raise your hand and we'll unmute you, and we will be able to let you ask your question. So I'm going to give people just a minute to type that in. One thing that was really funny to me was that you were saying about just being able to get a new species. We only recently in mine were able to get a wild caught species to add into our Bergia, which is T-Sleg line. So I feel for you. I hope you get one soon. It's even a generation away, though. I'm very happy you were able to.
Yeah, no, it's huge, and it's completely changed the landscape, because when I first, got here, I bought a bunch of super inbred fish, and I was like, well, maybe I'll get lucky and it'll be fine.
Yeah.
But they just never bred for me, really. And the couple of clutches that I did get from them were not great. Whereas now like I said, I'm starting to get some babies coming out of the F1s, and it's been really exciting to start to see them breed.
That's good.
Yeah.
Yeah, it's a relief after your first generation. Okay, we have our first question here. So how does the individual know biologically that there are predators or lots of siblings? What is the message that triggers the change?
Yeah, hi Lynette, by the way. I wish I could see you in person. Thank you for asking. That is a great question. So with the Daphnia, so that they're picking up on the predators in the environment, one of the main cues that they're picking up on actually is the hormones that are sort of released by the predators, so they can kind of pick up those chemical cues directly from the environment and incorporate that into their phenotype. And so I don't know exactly the specific change that the spadefoot toad tadpoles are picking up on with the siblings and the density of other animals in their environment. But again, I would imagine it has something to do with like pheromones or hormones that they're sort of picking up on from the environment.
Okay. So another question is, how exactly, so you're getting some of your species from the aquarium trade. Is there, like, zebrafish I know can't be, but where are you getting your other species? Is it all aquarium for the most part, or are you able to get some wild caught on some of the other species?
Yeah, so with cichlids, I mean, barring actually going over to Malawi yourself and catching them from the lake, you're really relying on, there's basically only one or two people importing. So all of your fish are channeled through that kind of bottleneck. So yeah, most of what we have is from that source. There are a couple of people. Actually, this is a crazy story. I picked up some F1 animals from a kid, for lack of a better term. I think he was in high school. I'm pretty sure he was in high school, who was breeding cichlids in his parents' basement in the northern part of New Jersey. So my husband and I drove to go and pick up these animals and do like a cash exchange with this kid. It was a weird,
science,
bizarre experience. Yeah. But he also was getting those wild caught animals that he was then breeding from people who are importing fish for the aquarium trades. It all traces back to that same sort of source.
So another question is, do you consider the similarities in morphologies of the pelagic versus benthic fish to represent an example of convergent evolution? I am not sure if I said those words right. So
it's a good question. It really depends. So with stickleback, I'll answer with stickleback because they're easy and familiar. There are stickleback that are independently derived that are all benthic. And so yes, I would consider that to be an example of convergent evolution, where you have this independent, you have a single ancestor that goes out and colonizes a whole bunch of different lakes, and by chance, many of them wind up looking benthic, but they're not benthic because their common ancestor was benthic. They're benthic because they encountered that condition in the environment and changed themselves over evolutionary time.
And can I ask my own question, just to insert, what is pelagic versus benthic? Because I'm not aware myself.
Yeah, so the benthic versus pelagic. So
that's right. It's right on the screen. I'm looking at you.
Yeah. You're a
terrible person. Concentrating too hard on the speaker.
Narrow focus, yeah.
Narrow focus. All right, thank you. That helps. All right, do we have any last minute questions that are better than my pelagic versus benthic one?
Hey, it's good to go over the basics. It's important. This is complicated stuff, so it's good.
All right, I think we're going to wrap up this one and move on to our last vignette. All right, thank you everybody for all the questions. Okay, so to start us off, we mentioned that you were doing some teaching as an addition to your research. And as we talked about, that's kind of an important part to your career as well. So what got you excited about doing science and teaching science, and why is that an important focus for your career?
Yeah, so I've honestly, almost as far back as I can remember, I have always loved teaching. And I can say that I started my PhD with sort of the explicit goal of becoming a teacher or an educator and not necessarily a researcher. I've told this to my PhD advisor. I fell in love with the research in his lab a lot more than I ever expected to. I really thought it was something that I had to do a PhD in order to teach science at a college level. So that's what I was going to do. And I had to do research to get a PhD. So that's what I was going to do. And I was just going to check that box and it was going to be done. But I wound up really enjoying what I did on a day-to-day basis for research. And so I sort of bonus, I get to do both now, which is really awesome. And for me, that love of teaching always comes back to summer camp. During, as I was growing up, I went to a summer camp, and during high school and college, I taught sailing at that camp for many, many years. And it was hands down sort of the best job that I've ever had. And I've been really lucky in that I've actually enjoyed most of my jobs. But when I was at camp, I really loved working with younger people. I really enjoyed passing down my skills and my knowledge to them. And it gave me and my friends an excuse to hang out on boats all day. So that was kind of a win-win.
Always a plus.
And yeah, it's always a plus, right? But while I was studying for my undergrad degree, I hit this kind of personal crisis because I'd entered college thinking that I wanted to go to med school because I liked science and I liked biology in particular, and I was good at science and I was good at biology in particular, and I felt like being a doctor, a medical doctor, was the only career option I really had as a biology major, which I think is a common story for a lot of people who do their PhDs. It's like we didn't really want to be medical doctors, but we didn't know what else we could do in science. So for me, it took about 2.5 years, but I really realized that that was not something that I was interested in. I didn't want to be a doctor, at least not that kind of doctor. I'm not good at witnessing other people's pain. I really, really did not do well at organic chemistry in particular. And I was kind of just struggling. I didn't know what to do next. And fortunately, I had this really amazing undergraduate advisor who unfortunately passed away this year. But she saw that I was floundering and she pointed me in this new direction, which was academia. She told me, you know, you can make a career out of teaching science and I think you'd be really good at it. And that was a lot more appealing to me because it combined two of my passions and it left out having to deal with other people's medical trauma. And so I was really excited when I started my PhD about the teaching side of it.
Awesome.
And as a grad student, I was pretty surprised by how little training graduate students got in teaching, considering that most of us were doing a PhD to become faculty, and faculty are educators, right? But that's not something we ever really get trained in as, you know, graduate students in science. They kind of just throw you to the wolves, right? It's like, welcome, hello, here's your mandatory 3-hour training from HR and how to be a good TA. Okay, now get out there and go be a TA. And at UMass, I think we were really lucky because there were a lot of opportunities out there available at our campus to get education training and training in education. But in order to know about those opportunities, you had to have an underlying interest, and you had to find the right training opportunities given by the right people. And you had to have the advisor who was really willing to let you take away your time from research to attend those trainings. And so that's a lot of ducks to line up in order to get an education doing the thing that you were supposedly being trained for. So I did get a lot of great teaching experience and a lot of great training and teaching under my belt at UMass, but that was one thing that I felt was actually kind of lacking overall in graduate education.
That's fair.
And when I was looking for that next funding opportunity, so after my postdoc funding for my first project ran out, I was reaching out to a couple of different potential advisors, one in particular that I've wanted to work with for basically my entire career. And he suggested that I look into this Inspire program at Rutgers.
You told me a little bit about that and how that was pretty key. Is that is that tied in with kind of some teaching stuff as well as some research funding?
Yeah, it is. That's the really cool thing about it for me. So the NIH has these funding opportunities through what they call the Institutional Research and Career Development Awards. And so those are handed out to faculty members all across the country. And they're just aimed at producing better, more well-rounded researchers and educators. So specifically, they're trying to increase diversity and equity in the biomedical sciences. And so to do that, they feel like they need to have educators who are better trained at educating.
That's fair.
So my current advisor, Tetsuya Nakamura, he didn't have funding for me through like his traditional own grants, but he suggested that I should apply for this Inspire program at Rutgers because it would cover my salary and my benefits for a few years. It would give us a chance to apply for some additional funding together. And I was really, really excited at that idea because it did tick so many of my boxes. It gave me more training, more formal training in teaching. It allowed me to sort of think about and talk about diversity in STEM. And it gave me basically 3 years of as much as any kind of funding opportunity can be guaranteed funding.
So how is this a little different? So this sounds a little different than like a normal postdoc experience. Can you do a little compare contrast for us? Because you've had two now, one without, one with. So
yeah, absolutely. So it is kind of night and day for me. So in my first postdoc, right after I defended, I was working with a main advisor at the University of California, Riverside and a collaborator. And all I did all the time was research. It was mostly field research. So I was especially kind of isolated, even more than other postdocs seem to be, because I didn't actually get a chance to meet most of the undergraduate and graduate students in my own lab, because I was not on campus for most of my time. And so for me, that was kind of isolating, and I didn't have that social support from a cohort of peers who were also going through similar things. It was kind of just me. I was lucky because the field station that I was at had a really amazing community attached to it. But it just was so different from like being in a graduate program where you have that sort of institutional support and peer support. And I knew at the time when I was doing my PhD that I relied on that support, but I don't think I really recognized how important it was to my mental health and to my productivity until it was gone.
Awesome. But, or well, not awesome at the time, but awesome that you have that a little bit more now.
Awesome that I got that back now. Yeah. So it's great to be back with a program with other people who are going through the program at the same time as me. So there's about 5 other people in my cohort of postdocs. And then there are 2 other cohorts at any given time. So there's right now there's a cohort above me and a cohort that are just coming in.
Yeah, it's nice to have a little bit more structure involved in this, especially when you're doing such a training to try and really learn to be educators of like that sort of, that's good to have, that you have a community.
Yeah, and then the other thing in addition to the community is that there is that explicit expectation that we're going to be teaching, and there's scaffolding for training for teaching as well. And so I wanted to talk a little bit about that too. So the salary split, 70% is research, and I told you a little bit about what I'm going to be doing for that. But 30% of it is that teaching component and also professional development. And so the teaching portion involves this semester-long course in scientific teaching, which is taught by one of the program leaders. And after we take that course, we're actually paired up with faculty members at one of 3 partner institutions. So that's New Jersey City University, Medgar Evers, and Wayne Patterson. And so these are 3 schools that are relatively close by Rutgers geographically, but they're different from Rutgers in that they're all predominantly minority serving institutions. And they have much more of a focus on teaching undergrads as opposed to performing research. Like Rutgers is very much like UMass, it's a research intensive school, whereas these 3 partner institutions are much more focused on teaching. So we go there and we learn teaching from our teaching mentors, and we give the undergrads a chance to interact with these cutting edge researchers nearby. And so the idea is that it's supposed to be an enriching experience for everyone, right? Because the teaching mentors get some experience sort of mentoring postdocs and teaching them how to educate students. The teaching mentees get, some experience teaching. And the undergrads get that chance to interact with researchers that they wouldn't otherwise have access to. And the fellows actually get some funds as well for mileage reimbursement and parking and stuff like that. So it's not like we're paying out of pocket to go teach. We actually have that included as well.
And I know we've got a couple grad students on the line at least. So how does one get involved with like an Inspire program?
Yeah, so I'm gonna, and I can share my slides later as well. Sorry, skipping through all the research stuff that I didn't have time to talk about. Oops. Here's what I wanted to show you. So these are the Inspire Fellows Twitter page that I actually run because science communication is a thing that I like.
You can definitely share that on, if you'd like, we'll share it on our website.
Yeah, that would be great. And so this is also the website for Inspire as well. And so one of the things you can do as a grad student is apply to come work with us. We have new cohorts starting next year. And so applications are usually due sometime in the spring. So you can start to think about, if there are advisors at Rutgers that you'd want to work with, this would be a great time to start thinking about that as a grad student. Yeah.
Awesome. Thank you very much. So 2 more questions, I think. We're almost done, but thank you for everybody who stuck with us. It's okay if you do have to leave, but we're going to just have a few more questions, then a question and answer at the end. So the first one that I want to ask you is, with the teaching and the research and all together, what are some of your goals going forward?
Yeah, I mean, I still very much want to be a faculty member one of these days. And so for me, my sort of short-term goal is to have funding to stay with my current advisor for as long as I need to find and secure a faculty job. So he and I are applying to some grants because, like I said, so the Inspire program is a 3-year program. And there's a little bit of flexibility, like the last cohort that was severely impacted by COVID, they gave an extra year of funding to just no questions asked. If you need to stay an extra year, it's okay, which was amazing, really supportive environment. But it is supposed to be a 3 years of funding, and I am now starting my 2nd year. So we are getting to that point, right, where we're starting to think about, oh God, the funding is going to be gone in another year. What do we do? So we're applying for some grants that would help sort of bridge that gap and give me another couple of years here where I can continue to work on some of this project that's just now really starting to really kick into high gear now that I have my animals, now that they're starting to breed, all that kind of stuff. And then after that, like I said, yeah, I really do want to be a faculty member somewhere someday.
Awesome. Yeah. And then my last question for the night is, I know something that's important to you is advocating for diversity in science. So what are some ways that you feel people can encourage to advocate for diversity in science and STEM? Because it's something that's important to us as a group, and we always want to hear people's perspectives on this because we think it's important to talk about that.
Yeah, I think, the biggest piece of advice I can give you is to just keep pushing. Support ways of reporting bad behavior and bad actors that don't rely on a vulnerable person or group of people reporting that behavior, because that's never a good way of changing anything. It's a good way of making sure that changes that need to be made stay sort of under the radar. I would also say, and again, you're going to have to balance this with self-protection, but don't be afraid to challenge your admins. I really thought that I was, given my involvement with graduate women in STEM at UMass, and how sort of vocal and outspoken I was through them, I really thought I was going to be lucky to get through grad school without actually being thrown out. But not only did they let me graduate and give me my PhD, the admins that I was actually most outspoken against really helped me cut through some of the red tape when I was having trouble onboarding at my first postdoc because of paperwork issues. Someone that I had repeatedly sort of challenged for being soft on diversity, equity, and inclusion actually personally did me a huge favor to make sure that my gap in pay didn't stretch over months, was limited to just a couple of weeks.
Yeah.
So I know it can be really, really scary to be vocal when you're in a vulnerable position. I absolutely felt that many, many times throughout my graduate career. But sometimes it can actually work out in your favor because the people that you're speaking out against might be privately cheering you on. And I think that's kind of what happened with me. Yeah. That said, of course, protect yourself, because you're never gonna be able to affect change if you don't, get to stay in where you are.
I understand. Yeah.
So it's definitely a balance.
Well, thank you very much. I am going to open it up for one more question and answer. We've got a few more minutes, so we can take one or two if you would like. So once again, down at the bottom, either the question and answer, or feel free to raise your hand and we will unmute you. One quick question is, do you have a favorite fish you like to work with? Is there one that you feel like is like annoying? You're like, oh God, why did I choose this? Or
okay, so I have a couple of fish that I got that were not species that I was like trying to get, but I just bought a couple of extras for, tampering aggression. One of the things with cichlids is that they have these really crazy social hierarchies. And so if you have too small of a group of fish in one tank, then one animal is always going to be picked on. Whereas if you can add a couple of extra fish, you can kind of spread out that aggression a little bit, and then it's a couple of individuals being picked on. So in order to do that, I got a couple of extras that were slightly smaller at the sort of start. And they were not the species that I was sort of aiming to get. And some of them are really, really pretty. So I have these really gorgeous, like electric blue, neon colored cichlids, and I absolutely adore them. And I told my husband we're taking one of them home with us after all this is done.
I understand. All right. I think we're going to wrap it up here. We only went over, if anybody has any last minute ones, raise your hand now, or, okay, we're good. I think we're good. So I would just like to do the end of the announcements really quick. So real fast, look out for emails for next semester's events. Thank you all for joining us. We're going to be continuing our 10-year anniversary next year with some familiar faces and a brand new type of cafe that we're excited to bring you. So check out our website, OEB Life Science Cafe, for more information. You're also going to find information on Dr. Dina Navon there. We're going to share some of the information that she's provided with us today, as well as we're going to highlight her past cafe if you want to hear more from Dr. Dina Navon and see what she was talking about back then. And then finally, if you have not remembered and you have a time, can you just scroll back and sign in with that virtual sign-in sheet? It would really help us to bring the cafes that you guys want to see. So finally, Thank you very much, Dr. Navon. This was amazing, and I really appreciate you coming to talk to us again and updating us on where you've been.
Hey, anytime. I really enjoyed being a part of this organization as a grad student, so it's great to be back as an alum. So thank you very much for having me.
No problem at all. And we will see you all next semester. And I'm going to stop the recording there.